So hi everyone, I hope that you can hear me. So my name is Shiran and I'm the head of the scientific program at Neuroscience Therapeutics. And today I would like to share with you key elements of our work. But first, I would like to thank the organizers for giving us the opportunity to present our work and for a really fruitful conference. So thank you for that. So Neuroscience was established 5 years ago with one major goal: to find an effective treatment for patients with ALS. And since inception, we advanced from research into preclinical studies and into the clinic, which we concluded with very promising results. And we are now spearheading the next phase with ALS and also exploring into new indications. So although we all know the numbers and the huge unmet need of ALS, it's actually the people behind these numbers who are really the focus here. And the person you see in the photo here is Shai Rishoni, and the impactful meeting between him and Alon, our CEO, is actually what led to the establishment of Neuroscience. And our drug PrimeC is actually a tribute to Shai, as his last name, Rishoni, is translated into prime in English. And so, as we know, despite of the huge effort of the field in the past 15 years, there are only 2 FDA-approved drugs that are available to date. And as we all know, almost all the phase 3 clinical studies have failed. And we believe that it can be due to the fact that they were only aiming on one target in a very complex and complicated disease. And therefore, we believe that the right approach is to tackle multiple fronts utilizing a combined therapeutic strategy. So Neuroscience flagship drug for ALS, PrimeC, is a unique formulation consisting of 2 FDA-approved drugs, ciprofloxacin and celecoxib, that are aimed to work together synergistically on more than one ALS target. And just to drill it down a bit, we know that the pathophysiology of ALS is very complex, and it underlies multiple pathways and mechanisms that are involved. And therefore, we believe that the right approach is to tackle multiple fronts. So with PrimeC, we aim to target iron accumulation together with dysregulation of microRNA and neuroinflammation, all, as we know, considered key hallmarks of ALS, and all eventually leads to neuronal death, in which each one of the compounds targets several of these pathologies. But importantly, the unique extended release formulation of PrimeC really allows us to maximize the synergism between these 2. So once we had PrimeC and the formulation in our hands, we were able to move forward into the preclinical studies, of which we conducted with Zebrafish model. As you know, Zebrafish are a very successful model for drug screening. And so we conducted our studies on 2 key models for ALS. One is the SOD1 mutant that was conducted in Israel under the supervision of Dr. Niva Rosak-Blum. And the second is the TDP mutant that was conducted in McGill University under the supervision of Dr. Guy Armstrong. And what we found in our preclinical studies was really outstanding. We saw an improvement of 84% in the distance performance of the Zebrafish, representing the motor performance, and 110% in the TDP-43. And we also saw a recovery in the motor neurons together with the supporting cells, the microglia. And as they say, a picture worth 1,000 words. So I thought it will be nice for you guys to just see it for yourself. So you can see in the middle panel, it's the swimming pattern of the zebrafish. So you can see the healthy one swimming really nicely. And compared to that, we see the ALS zebrafish with really impaired activity. And following treatment with PrimeC, we gain a pattern which is very much similar to the healthy ones. And as you can also appreciate from the graph on the left panel, we see clearly the synergistic effect between these 2 compounds and the dramatic effect of PrimeC, especially relative to the standard of care, to riluzole. And this was very encouraging and actually led us to dive even deeper into the synapses and in order to try and understand this effect of PrimeC. So as you can see here in the right panel, you see the neuromuscular junction. So we see the presynaptic compound in red and the postsynaptic compound in green. And when we have colocalization, we see yellow. So as you can see, we gain much more yellow following treatment with PrimeC, indicating more intact synapses and less orphan pre and post compounds. And this was also reinforced by looking on the morphology of the neurons, thanks to the fact that the zebrafish are transparent. So we were able to see actually the morphology of the neurons. So you see the severe xenopathy of the ALS zebrafish, and compared to that, following treatment with PrimeC, we gain a morphology which is very much similar to the healthy ones. So these results were very encouraging and actually suggested that there is maybe an overprotective effect here to PrimeC, and these results, together with the supporting of our staff and leading clinician in the field, gave us the green light to move forward into the clinical phase, and this is exactly what we did. So we conducted a phase 2A proof of concept study in the Suratsky Medical Center under the supervision of Professor Vivian Drori, with 15 patients in over 12 months of dosing, in which patient came to the clinic every 3 months, and in between they were monitored by phone. The primary endpoint of the study was safety, and it was evaluated by AEs together with blood tests and ECGs. And we are also looking on efficacy as exploratory endpoint by looking on ALSFRS-R together with forced vital capacity. Additionally, we were also looking on ALS-related biomarkers, which I will show you. So when we first wanted to assess the safety profile of PrimeC, we were looking on and monitoring the AEs from the study, and as you can see here, we didn't see any serious adverse events that were related to the study drug. And additionally, we also didn't see any unexpected AEs, and the AEs that we did so was mainly gastro, and they were transient. And we next wanted to further assess the safety profile of PrimeC, so we wanted to use the prediction algorithms in order to look on the predicted survival of our patients. So we used the Urgent model together with the Enkels model, and as you can see, both models were in agreement that the patient performed as predicted, indicating an additional aspect of safety. And next, we wanted to assess the efficacy of this study by looking on our patient compared to placebo matched patient from the PROC database. So this work was, and actually all the analysis in this study was conducted by Dr. Ruben Van Eyck, and this is actually the time to thank him for that. So this was a propensity matching comparison based on 9 criteria, and as you can see, when we compared our patient to the PROC database to a matched patient from the PROC database, we saw a difference of 30% in the respiratory function in the FVC and 18% in the functional rating scale. And though it's not statistically significant, it is meaningful because this 18% represents actually 2 points. And as we all know, actually every point is meaningful because it's basically a function that is maintained. So following that, we wanted to further and explore the effect of PrimeC on ALS-related biomarkers. And to do so, we collaborated with a company called Neurodex in order to analyze neurons-driven exosomes that were extracted from blood samples. So basically, as we all know, exosomes are vesicles that contains representative content from the brain. They go in spontaneously into the bloodstream and basically allows us to measure parameters from the brain in a non-invasive way. And this is exactly what we did in our study. So first, this experiment was conducted in 2 phases. First, we collaborated with Mass General Hospital in order to characterize the basic phenomena of a cassette of biomarkers. So they generated us with samples of healthy subjects and patients with ALS. And once we established the basic phenomena, we moved forward into our NSC-002 study samples in order to assess the effect of PrimeC on these biomarkers. So first, we just wanted to characterize who are the main pathologies in play, who are the biomarkers that we're going to look on in our cassette. So of course, we turned to our mode of action and looked on markers that are related to iron accumulation and to dysregulation of microRNA using a biomarker for AGO2, which is one of the compounds in the complex of Dicer, as well as biomarkers that relates to neuroinflammation. And we also looked on TDP-43 accumulation that, as we all know, is a huge marker in terms of ALS pathology. And we also looked on two biomarkers that relates to cellular processes. One of them, LC3, is related to autophagy. We know that in ALS and also in other neurodegenerative diseases, there is impairment autophagy. And this is the reason that we chose to look on this aspect. And we also looked on cathepsin D, which is a marker for lysosomal trafficking. And we know, same as in autophagy, that the lysosomal trafficking is impaired and it's dysregulated in ALS. So we also chose to look on that. So when looking on the result from the first phase, when we compared the samples of the patient with ALS to healthy controls, you can see that we gain higher levels of biomarkers that represent microRNA together with TDP-43 levels and iron accumulation. And we also saw a similar trend in neuroinflammation. And this was also accompanied by lower levels seen in LC3 and cathepsin D, representing impaired autophagy and lysosomal dysfunction. And importantly, when we looked on the effect of PrimeC on these biomarkers, we were able to see that PrimeC was able to attenuate the ALS-related pathologies that we've seen in the previous phase. So if we are talking as an example about TDP-43, so we and also the literature saw that there is higher levels of TDP-43 in patients with ALS, and it's actually, according to previous study, increased with time and with the progression of the disease. And here we see that following treatment with PrimeC, we gain a longitudinal decrease in the levels of TDP-43, and this was also accompanied to a decrease in neuroinflammation and attenuation in the cellular biomarkers that we were looking on. And lastly, we wanted to assess actually the connection between these biomarkers that we see and the clinical outcomes. So we started to do some correlation between these two. So this is just one example. So you can see the changes from baseline in the levels of TDP-43 comparing to the deterioration according to the ALS-FRSR rating scale. And as you can see, in patients in which we were able to lower the levels of TDP-43, we were also able to attenuate the deterioration of the disease, meaning that there might be a biological activity here to PrimeC. And lastly, due to their importance in the field, we were also looked on neurofilaments in collaboration with Biogen. And as you can see, we didn't see any significant change following time with PrimeC, but we do plan to further explore this aspect in a placebo-controlled study. So to conclude all that I said till now, we know that we can conclude and say that this phase 2 a proof of concept study showed very promising results as the study endpoint was met and PrimeC was found to be safe and tolerable. We also saw encouraging clinical signals together with significant changes in ALS-related biomarkers. And so based on these encouraging results, we are now planning to further expand our biomarker experiment and continue with the development of PrimeC with a large placebo-controlled study in order to assess the effect of PrimeC for ALS. And so I would like to thank all our collaborators and colleagues for their great help and work, and of course you guys for listening, and I'm welcoming you to ask any question you may have. So thank you.
Thank you very much. That's really very, very remarkable. And what I think is incredible is the amount of progress that Neurosense has made over the last 18 months. That really represents huge benefit. So let me throw open this talk to any questions. One that is asked is why the did you compare PrimeC to edaravone as well as riluzole?
No, because if you're referring to the preclinical study, so we can give Radicava to Zebrafish, so we were only able to compare to the riluzole.
Okay, thank you, thank you. So let me ask you. So among other things, celecoxib is a COX-2 inhibitor.
Yeah. Did
you look at the overall levels of COX-2 activity in patients? Were they elevated? If not, I presume that would emphasize the other mechanisms of action of the celecoxib.
So yes, we did look on the levels of COX-2 together with all the downstream cascade of celecoxib. We are still analyzing this data because, you know, it was an open label study, so we can't, unless it's really decisive, it's really hard to say, but we didn't see a elevation in the levels of COX-2 in our patients.
Yeah. And another question that I've wondered about is, are the doses of each of these that you use more or less the kinds of therapeutic doses we would use taken one by one in patients?
So the doses that we use are unique doses that you can't get from just pharmaceutical compounds, but we don't the levels of each one of the compound doesn't go up more than the levels that are known.
Thank you. Okay, well, that's again, congratulations on making such great progress. Let's turn now to the next talk, which is from Spinogenics, entitled